Hello!
Let's talk bacteria.
Bacteria are single-celled microorganisms found everywhere in our environment. They are extremely small and diverse living beings that play a crucial role in ecosystems and human health. Studying bacteria and understanding their growth can provide us with valuable information about their function and behavior.
Dishes that receive no light. 3 of them are agar, which have 30 hours, and 2 of them are a gelatin medium, which have 12. These are the petri dishes that are under direct sunlight, and the agar plates have 30 hours of being here, and the gelatin medium have like 12.
This is June 18 with D2's bacteria on room temperature without direct sunlight in a sealed box. These are 2 in agar, this one in the nebulator mask. We can see the lines but almost no bacteria. And this is another one, which is the bacteria on the keyboard, and we can see some changes from yesterday to today.
Hi, I'm Miriam, and I was in charge of testing the susceptibility of different bacteria to natural essential oils. So here I'm going to show them to you, and I hope you enjoy this experiment and that you have learned along with us. These are the Petri plates that we grew on day 1 and day 2. However, there are no visible bacteria on the agar plates, and as you can sort of see, they have dried up. The agar is very thin-like, and it is a very plasticky material. I'm going to show you an example that we set out to dry with no bacteria on it, so you can see the texture of it currently. This is the texture of the agar dish that failed. As you can see, it is very thin-like. So as you can see, day 1 and day 2 were completely a fail. Now, these here are LB agar plates from Amazon, and we took bacteria samples from my house and swabbed them onto the agar plates, and bacteria did grow here. This was on June 16th of 2023. I'm going to show you the bacteria that grew here. So here we have my mouth, my dirty mouth before brushing my teeth. So that's how it looks like. Then this is the air conditioner. This is another sample of my mouth before brushing my teeth. This is my hand. So basically what I did for this, because I am not a dirty person, I dragged my hand along the floor, and this is what grew. That's a lot of bacteria. Then here we have my school shoes. Over here, the kitchen handle. And the kitchen sink. Okay, so now I'm going to show you the LB agar plates where we tested the inhibition of bacteria. So here we actually know for sure that the essential oils killed the bacteria, and it's not that our Petri or our agar plates were failing like here. So let me show you a comparison of the control to the sample of bacteria that we swabbed, and we added drops of essential oil. So here is the floor sample. So this is how it looks. As you can see, there is no bacteria. This over here is just some dirt from when we swabbed it, but no bacteria grew. Here are the drops of our essential oils. This one kind of dragged down a little bit. Here are our drops of the essential oils. This one spilled over a little bit. So this is the floor without adding any drops of essential oil. So as you can see, there's a big difference. Here we have the bathroom handle. As you can see, nothing grew on the bathroom handle. We have no bacteria, just some drops of essential oils, as well as the toilet. Nothing grew on the toilet either. Just the drops of essential oil. And I'm pretty sure you can see sort of where I swabbed it, which is over here. This is where I streaked my sample. This proves our hypothesis, right? Which is that no bacteria would grow on the agar plates where we added our drops of essential oil. However, we did have a control, and bacteria was supposed to grow on the control, which is this little section over here, and it didn't grow on any of them. So we think that the essential oils evaporated, killing off the bacteria near, because the agar plate is very small and all of the sections were really close to each other. So that's why you can better see the comparison with these two agar plates, with the floor one over here, which will work as our control. Now, on June 17th, Alejandra and I decided to make more agar plates, but we changed our medium by adding beef bouillon cubes to the agar, or beef powder, and now the bacteria was visible to the human eye. Now, the first time, bacteria did not grow on our agar plates because we did not add beef bouillon cubes nor beef powder to our agar medium. We only added it to our gelatin medium, which did grow, but it liquefied, so I can't really show you that. By adding the beef bouillon cubes and the beef powder, we made a rich nutrient medium where bacteria did grow. These are the samples that we took on June 17th. So we have another sample of school shoes. Now, this sample grew fungi, so that's very interesting. Here you can see it. This is my mouth, now cleaner. So we only have a few colonies of bacteria, only 1, 2, 3, 4, 5, 6, 7, 8, compared to the other sample. Here we have clean hands. Those don't look very clean. Here we have the floor. So this is the sample of the floor that I showed previously. Some fungi as well. Now this is my mom's mouth. So we only have 1, 2, 3 bacteria growing on there. And this is my dirty hand. So let's look at the difference between those two. So as you can see, the clean hand did not grow any fungi, only a few bacteria, and the dirty hand grew much more bacteria. Now these are the last samples we took. So this is from my phone. So that's quite dirty. There's some condensation. This is from another sample from the floor on the same area that I swabbed this. So obviously it's much cleaner because I picked up a lot of bacteria for this plate. Then we have the shoe again. Again, much cleaner because I picked up a lot of bacteria for the other agar plates. And then the kitchen handle, which looks like this. Now, to conclude the experiment, I will be adding some drops of these natural essential oils to see if the bacteria will die or if it will survive. You can see the results tomorrow at the science fair if you come visit our stand. Thank you for watching, and remember, this is Miriam, over and out.